camera is in place, so we can start, right? That's good. Right, so the next couple of lectures I will be speaking about some stochastic models of population evolution. So this is like the second part now. And there are two classic models in population genetics that are called the Wright-Fisher model and the Moran model. And I will be discussing them in turn, and we'll see some simple properties of these models that we can derive. So let's start with the Wright-Fisher model. And so the idea is that we want to study the change of type frequencies in a population, similar to what we did on Wednesday, but now we want to do it in a stochastic setting. So we want to have a stochastic model which works at fixed population size. And so in the Wright-Fisher model, the picture is the following. So we have individuals. So we have n individuals. And they are labeled by some index I from 1 to n. And so let's depict each individual by a little dot here. So these are n individuals. And they have some types. So, they could all have different types, or they could be several of them with the same type. That doesn't really matter. And then we have a time, right? So time goes downwards. And so in the Wright-Fisher model, this is what is called a model of non-overlapping generations. So this means that in each generation, so non-overlapping generations, this means that in each generation, all the individuals, if you will, die and are replaced by other individuals. So this is not very natural for humans, but it would be a natural situation for example, annual plants which die in the fall, and then the next generation is created by the seeds that come in the next generation. At this point, we don't need to think about the biology too much. The main point is that it's a kind of discrete process. So we go from time t to t plus 1. And so in one time step, each of these individuals will leave a certain number of offspring, right? So basically we can think about this. So here we have the next generation, right? And this is basically the picture that I drew already on Wednesday. So each of these guys leaves a certain number of offspring. So for example, this individual perhaps leaves 3 offspring. These individuals leave no offspring, and so on. Okay, and this guy perhaps leaves 2 offspring, and this one maybe also 2. Okay, so this would be a possible scenario. And so what we need to specify is the statistics of these offspring. Okay. So let's say for example, let's say. So individual I leaves ni offspring in the next generation. And so this picture, for example, if we label these individuals by 1, 2, 3, and so on, this would, for example, mean that individual 2 leaves 3 offspring, so n2 is equal to 3, n1 is equal to 0, n3 is equal to 0, and so on. Right? So this is sort of how you should think about this. And this is essentially all that happens. The offspring, of course, inherits the type. So if the parent has a certain color, or a certain fitness, then this is inherited. So the offspring inherits the type of the parent. And this includes, of course, in particular its fitness. And for this whole discussion, we will not consider any mutation. So the types are passed on to the offspring, but they don't change, right? So we have no mutations. What this, of course, means, and this will become important in a moment, is that types can die out, but they cannot be recreated. So the diversity will decrease, so types can go extinct. But cannot appear, but cannot be created. And so this already, tells you that the stochasticity here will have a kind of an effect on decreasing the diversity, because types can only, the number of types can only decrease. Okay, so the diversity decreases. Okay, so this is sort of the general framework. Now we have to get a, we have to define the model more mathematically. So what we need, and what we're sort of looking for, so what we need is a kind of joint distribution of these offspring numbers. Right, so these ni, so there's some joint distribution T of n1 up to nn. And this joint distribution should satisfy the constraint that the population size should be constant. So we want that the total number of offspring should be equal to n. So this is what we, and this is basically what defines the model. So the Wright-Fisher model is defined by a certain choice or a certain construction of this offspring number. Is this clear so far? I should say also I have some handwritten notes, so if you want I can sort of distribute them over the email list after the lecture. I mean, it will sort of depend on a little bit of what I exactly do, but I can sort of put together the most important things, so then you can sort of check. And maybe since you weren't here last time, maybe you want to write down your email here? Yeah, okay. Okay, so this is the idea, so let's now try to make this more formal. So what one does in the Wright-Fisher model is to assume that the offspring number, to begin with, the offspring numbers are Poisson distributed. Okay, assume that these ni are Poisson distributed. And so for each individual, there is a Poisson distribution, and it has some mean, and this mean I will denote by Wi, and this is the fitness. Okay, so the fitness of that individual is the mean number of offspring. And so that sort of corresponds to the RIs in the deterministic case that we had on Wednesday. So then, if this happens independently for each individual, then of course the joint distribution is just, so this means that, okay, let me write down the Poisson distribution, so the probability that the ith individual has ni offspring is 1 over ni factorial e to the minus wi times wi to the n. Right? So this is the usual definition of the Poisson distribution. And if this happens independently for all the individuals, then of course the joint distribution is just the product. So if these things are independent, then the joint distribution is just the product of these things. And so we can write this, so this will just be the product. Right? And so this is equal to, and here, the exponentials lead to a sum of the fitnesses, which I can write as e to the minus nw bar, where w bar is a mean fitness, and then I have a product I equal to 1 to n wi to the power ni divided by ni factorial. Okay, so this would be,
capital N is the number of the species or the
sorry?
Capital N is the total number
It's
of
the number of individuals. The number of individuals.
And also the number of the species.
No, what I'm saying is that individual I has fitness wi. Of course, many individuals can have the same fitness and can be of the same type. I'm not specifying that at the moment. I'm just saying each individual has fitness wi. Okay? So the number of, I'm not talking about the number of types here. I mean, this is basically, each individual has a type which leads to a certain fitness, but many, of course, individuals can have the same type and have the same fitness, but this doesn't really matter on this level. Okay, so later on we'll basically consider the case when only 2 of these W's are the same. But at this point, n is really the number of individuals, and W bar is, again, is the mean fitness, so this is 1 over n times the sum. Okay? Now, the question is, so how do we now constrain, how do we make sure that the number of individuals doesn't change? Now, if we,
No, okay.
You think it's okay? Okay. So if we define the model like this, so we say each individual creates offspring independently, then this would be something that is called a branching process. Okay, so I don't know, who knows about branching process, has heard about? So we'll talk about branching processes a little bit more on Monday. But so this would lead to a branching process. Now the question is, how would you, can you make sure that the number of individuals doesn't change? If you do this independently, then of course the number of individuals generally will change, right? So under this definition, this will lead to a so-called branching process. And so then the number of individuals will not be fixed. Individuals in the next generation is a random variable, all right, so I call this RG, so it will be a random variable, and prime, which is just equal to the sum of these guys. And in general, this would not be equal to n, of course. Now you could think, okay, maybe we can make it be equal to n on average by saying that the mean fitness should be 1. Okay. So if we impose that the mean fitness is 1, then you might think that on average the number of individuals shouldn't change. But the theory of branching processes tells us that if we do that, then the population will go extinct with probability 1. I mean, the problem is that you cannot suppress these fluctuations by just fixing the average fitness, and the process will generally go to extinction. So this will not work. Okay, so fixing the mean fitness equal to 1 will then lead to extinction. And in fact, any value, so this is something that the theory of branching processes tells you, that any value of the mean fitness that you impose, the population size will either go to 0, so it will go extinct, or it will grow exponentially. So you cannot, within a branching process, you cannot control the population size. Okay, so n cannot be controlled. So one has to do something else, and what one does mathematically is to condition the population size, right? So this basically, the way you can think about this is that you run many realizations of the process, and you only keep those where the population size is fixed. Of course, this is always a possibility, and you only keep those, so that means that you condition on the population size being constant, and then you get an expression for the joint probability that is reasonably simple, which we can write down. Let me write it down. Maybe we don't need all of this at the moment. Okay, so what we'll do is instead of doing this branching process, we'll condition on n prime, so the population size in the next generation to be equal to n. And so for this one needs, of course, the probability of this event, and this I'm not going to derive, I mean that's not very difficult. It can be shown, so first of all it can be shown that the probability, so maybe I can get rid of this picture as well, we can get a new one later. That the probability, so I always use for probability of events, I will use this notation with a fat p. So the probability that n prime is equal to n, so the probability that the next in the next generation the population size is the same is also given by a kind of Poisson-like expression, which looks like this. And so this you can just derive by, so basically what you need to do is you need to sum these joint probabilities under the constraint that the total population size is a constant, and then you get this expression. And so therefore the conditioned offspring distribution then becomes of the following form. So this is now the probability of these offspring numbers conditioned on capital N. And so this is just, of course, equal to this product here of the independent events divided by this thing up here. Right? you put these things together, what you find is just a kind of multinomial coefficient. So this is a multinomial coefficient times the product. So this is what comes out. And so what this actually is what is called mathematically multinomial sampling. Okay, so what you actually do is multinomial. And so informally, what this means is that you simply So the way you sort of the easiest way to imagine this is that so you want to create the new population from the old population. Of course, what you know is that each individual has to have a parent, right? So if we draw this picture again, and we have here the generation T, and here we have the generation T plus 1. Right. So each individual has to be connected backwards in time to one other individual. And so what this multinomial sampling formula simply means is that you have to choose for each individual that has to choose a parent, and the probability of choosing a parent is given by its fitness divided by n times W bar. So basically, you look at this individual, so these guys all have some fitnesses, right? W1, W2, W3, and so on. So you pick one of them as a parent, of course, this order here doesn't really matter. So strictly speaking, of course, you could also, pick this guy as a parent, and maybe this one will pick this guy as well, and so on. But each of these individuals has to pick at least one as a parent. Okay, maybe these three all pick this one as a parent, and then you have the new generation. Okay, so informally this means that each individual or each child picks its parent with probability Wi divided by n times W bar, which is just equal to Wi divided by the sum of the fitness values. In this way, you have then created a new generation, and then again, each individual inherits the fitness from its parents. So this way, you can just propagate this process. Okay, so this is the Wright-Fisher model. Okay. Okay, so now to actually do something with this, we have to of course consider simple cases. And so the simplest situation that one can consider is again of two types. So we would just look at two types, and we moreover assume that there is no selection. So the fitnesses are the same. Of course, when the fitnesses, if the fitness of all individuals are the same, then it's just 1 over n, and then you just pick your parent at random. Okay, so now we consider the following simple case. Guys, I think I can, yeah, okay. So we have two types. No selection. So this is what I call the neutral case on this thing. And of course, if there are two types, we can call these types again A and B, and then we have NA individuals of type A and NB of individuals of type B, and the sum of the two is equal to capital N. And so in fact this then the model has in some sense only one degree of freedom, but so we only need to consider the number of A individuals, which I will denote by small n. Okay, so consider and so the number of A individuals can take values from 0 up to n. So this is what we want to consider. So essentially we have a kind of Markov process on this state space, which are the integers from 0 up to n. And this Markov process is characterized by transition probability. So what we need is a transition probability which I call T. And so this is the probability that the number of A individuals is n prime in the next generation if it was equal to n in the previous generation. Right? So this is the basic quantity that we need to consider. And now, of course, everything is sort of random, so this is just the probability that when children choose their parents, that the A type was chosen n prime times, right? So there's a probability that an A individual is chosen n prime times in any order, of course. And so this will, of course, be equal to the binomial coefficient n over n prime. So this is the number of ways in which these n prime individuals can be selected among the n individuals. And then we have to select, now the probability to select an A individual in one step is just n divided by capital N. Right? This is the probability, since there are n individuals in the previous generation, the probability of choosing an A individual is n over small n over capital N, and this has to happen n prime times, and the other choices have to be for a non-n individual, a B individual, and this will happen with probability capital N minus small n divided by n to the power n minus n prime. So it's sort of important so the probability of choosing an A individual is just small n over capital N, and the probability of choosing a B individual is 1 times that, right? Right? So the n is here and the n prime is up there. And so of course in principle in this, and this is sort of what makes this model, as we'll see, relatively complicated. In principle in one generation, all the A individuals could go extinct, or all the B individuals go extinct. So you can have rather large, with small probability, you can have rather large changes in the type composition in one in one step. So this basically, so now you can start your process somewhere here, and then you do transitions according to this transition probability, and this will sort of jump around on the state space. And the question is, what will happen? Okay, so what will happen in the long run? This is maybe not so difficult to guess. So what will happen to this process if I wait long enough? Where will it end up?
Extinction or one.
Right, so one of the two types will eventually go extinct. So this means that the process either goes here or goes here, right? So these are, because we have no mutations, the only possible outcome is that one of the types goes extinct. So if you will, the long time behavior is extinction of one of the types. And this is also in a sort of in statistical physics jargon, one would say that the states n equal to 0 and n equal to capital N are absorbing states. And this, you can see, from this formula, of course, that, for example, if n is equal to 0, then for any non-zero n prime, the transition rate will be 0, right? So if n is equal to 0, then the only way of getting a non-zero transition rate is to have n prime also being equal to 0. So this is equal to delta of n prime 0. And similarly, if n is equal to capital N, then the only possible state is that n prime has to be equal to n. Okay, so this is clear. So the process will sort of jump around, but eventually will go to extinction. And so the extinction of one of, and this is sort of different from, qualitatively different from what we have in these for these infinite populations that we discussed on Wednesday, because there we have differential equations, and these differential equations, of course, asymptotically can approach a state where one of the populations is almost dominating, but you never really, strictly speaking, hit 0. So you don't really have extinction in that sense, but here this will happen. And this is then called a fixation. So I mentioned that already on Wednesday. So so we have these sort of so this extinction means that the surviving type then goes to fixation. The surviving type goes to fixation. So the question that we want to answer here is the probability of fixation. So the first question we want to answer here is so what is the probability and let's say probability of fixation of type A if there are initially small n individuals of type A, small n individuals. Right, so suppose I start my process at some point here, and then I let it run, and then it will hit this point with some probability, or it will hit this point with some probability, and this is a fixation probability. Okay, and this is sort of the first important question that one would like to ask. And in this simple case, of course, this is not very difficult to answer, and but I think it's sort of instructive to make a little formal argument to derive it. For that, we basically only need this transition probability, so I think this here. So this is the question now. So the argument is based on considering the following. So we can ask, conditioned on the initial point. So suppose I start at some value n here. What is the expected value of a individuals in the next step? Okay. So what is sort of so given n individuals, so a individuals. At time t, what is the expected value of n prime at time t plus 1? So let me write this in sort of mathematical jargon or notation. So this is the symbol for the expectation value. So what is the expectation value of n prime conditioned on n? Okay. Now obviously, since this is just binomial sampling, this will be equal to the probability of choosing an A individual times the total number of individuals. But the probability of choosing an A individual is of course simply n small n over capital N. So this is equal to n, okay? And so this process has the property that, independent of where you start, the expected number of A individuals in the next step will be the same as it was before. So this is a kind of symmetry property, and this is called, in mathematics, this is called the martingale property. So who has heard about martingales before? The mathematicians have.
No, I know the expectation is see at the mean in the
right, yes. So this is something that one uses often in random walk theory and so on. So this is the martingale property. So the expectation after one step is the same as the initial point. And so this one can immediately use to deduce the fixation probability in the following way. So the expected, the expected value of n prime after fixation. And so what I know is that this average doesn't change. So I start the process somewhere, and I know that this average doesn't change. Now I let it run for a long time. It will either hit this boundary or it will hit this boundary with some probability. So the expected value after fixation will be just equal to the probability that, so if the type A type has gone to fixation, this will be, and I will denote these fixation probabilities by pi n. It will be the fixation probability times capital N, or if it doesn't fix, the number of A individuals will be equal to 0, right? So this will be equal to pi n times n. So pi n is the fixation probability so the probability of fixation of A type with n initial individuals. Okay, so this is equal. On the other hand, we know that this at all times this has to be equal to small n because of the martingale property. So we conclude that the fixation probability is simply capital N over small n. So, in particular, of course, if you start out with capital N individuals, the fixation probability is 1. If you start out with 0, A individuals, the fixation probability is 0, and in between it just interpolates linearly. Okay? And this is not very deep. I mean, another way of thinking about this is to say that you have in your initial state, you have capital N individuals. If you now think about the sort of genealogies of these individuals, right? These individuals will sort of leave offspring in the future, and since in the long-term future, all types, if you think about, for example, suppose you assign different types to all the individuals. I mean, if they have the same fitness, you can of course do that. It doesn't really make a difference. Then you know that at long times, only one type will survive. So this means that, except for one individual, all other individuals will have no offspring at very long times, right? So this is a common feature of genealogies. That if we go back a few, I don't know, 100 generations, then all of us will have basically the same ancestors, right? So long times in the future, only one individual will leave an ancestry, but since all the fitness values are the same, the probability that any one of the individuals will survive to infinite time is the same for all individuals. So if there are initially small n A individuals, then the probability that an A individual will be the surviving one is just small n over capital N. So that's more or less the same thing, but this is a kind of more formal argument. Okay, so this gives us the fixation probability in the neutral case. And maybe this is a good time to take a short break. Do you have some questions? Or you said it's a good time?
It's okay.
To take a break, right? So this is this of course is rather obvious. Now the interesting question that I want to discuss after the break is what happens if you now add selection, right? So if the two individuals, if the two types are not equivalent, so what we want to discuss is a fixation in the presence of selection, and it turns out that this problem is not, within the Wright-Fisher model, this has not been solved. Right? So this just means that instead of having this transition probability, you have to add the fitness values here, but this already becomes something that you cannot solve in closed form. So therefore, we will introduce the second model, which is called the Moran model. And there we can actually derive the fixation probability. Okay, so let's take, how long should we take? 10 minutes? Okay, let's take 10 minutes. Yeah. So the Wright-Fisher model is essentially, as I mentioned, it goes back to Wright and Fisher, so more or less it was invented in the 1930s, although I'm not sure at what point the mathematical formulation was really written down in the way that I did here, but this is a little bit later. So here the idea is rather than replacing all the individuals at once, you sort of replace one individual at a time. Okay, so here you have again a population of size n, and in one reproduction event you select an individual for replication. So in the first step, the individual, let's say I, is chosen for reproduction, so it just creates an offspring. With probability that is again Wi divided by n times Wi. So now we consider again general fitnesses, so you have some probability of being chosen. So now you have, of course, because this individual has been chosen for reproduction, so chosen for reproduction means simply you sort of you replicate this individual. So now you have two individuals instead of one, so the population size has gone up. And this has to be compensated in the next step. So in the next step, you choose a random individual to kill. Okay, so you kill a random individual. Another randomly chosen individual, so without reference to fitness, randomly chosen individual is killed. So if we draw this little picture again, then I go from t plus t to t plus 1. Then, for example, this individual here is, I decide to reproduce this guy, and then somebody else has to die. Right? So, for example, if I choose to kill this one, then all the other ones will just more or less stay as they are. Right? So this one is killed. So in terms of these offspring numbers, one individual has offspring, or has two offspring, one individual has 0 offspring, and all the others have 1 offspring. Okay. So now we want to consider again the case of two types. And now we will consider that these types don't have the same fitness, because we actually want to look at the effect of selection. So we have two types with fitness WA. I call this fitness 1 plus S, and WB has fitness 1. All right, so when S is positive, this means that the A type is preferred, so S is called the selection coefficient. That's essentially the same thing that we introduced also on Wednesday. Okay, and now we can again look at this transition probability. So what is the probability of having n prime individuals at generation t plus 1 if we have n individuals at generation t? Now in this case, of course, there are only 3 possibilities, right? So either you increase the number of A individuals by 1, you decrease them by 1, or it stays the same. And so this is easy to write down. So suppose that I pick an A individual for reproduction. This happens with probability, so according to this will just happen, any A individual for reproduction, this will happen with a probability that is equal to the fitness of the A individuals times their frequency. Okay, so this is the probability of choosing an A individual for reproduction. Then I have to choose somebody to kill. Of course, if I kill also an A individual, then the number of A individuals will not change. So I have to kill a B individual. And the probability for killing a B individual is just equal to the number of the frequency of B individuals. So this is equal to 1 minus the frequency of A individuals, right? So this is the probability to reproduce an A. And this is the probability to kill a B. And if this happens, then the number of A individuals has increased, and so we have n prime equal to n plus 1. Okay? On the other hand, if I want to reduce the number of A individuals, then I have to pick a B individual for reproduction. Which happens with this probability. And then I have, sorry?
N, small n is the number of A.
Yes, I'm sorry, yes. So I have to, that's right. So I have to, you're right. So I have to choose a B individual for reproduction. That happens with probability 1 minus small n over capital N, and I have to choose an A individual to kill, and this happens with probability capital N, small n minus capital N. So under this condition, n prime will be equal to n minus 1. And in all other cases, the number of A individuals will stay the same. And so the third possibility here is that if the other possibilities which we can just okay, so you can write maybe here. So n prime equal to n with probability 1 minus t of n plus 1, n minus t of n minus 1. Okay, so these are the three possibilities that we have. Okay. And now again, so the process is much simpler because we only have the possible changes of n are limited, right? So again, think about the state space here. And I start here, then I can only do nearest neighbor jumps, right? So the process can only go here or here, but it cannot make any longer jumps. So it's a kind of, so this is now really like a nearest neighbor random walk, but with a kind of position-dependent rates, okay? And so now again, we want to calculate the fixation probability. Calculate the fixation probability. Which I call IN, with n initial individuals. And this one can derive using a kind of recursive procedure. This is, mathematically speaking, this is really a kind of first passage problem, right? You have a random process, and you want to know what is the probability that this random process hits some predefined boundary before it hits another boundary. So it's a first passage problem. And for these kinds of first passage problems, one can often try to write down recursion relations, and this is what we will do here. So we're going to just look at how this pi n changes in one time step. Okay? So we will consider the first time step. Okay, so maybe let me draw this picture here again. So we start here, and so in the first time step, we either go to n plus 1 or we go to n minus 1. Right? This is all that can happen. And so the probability to go to fixation starting from n is equal to, well, there are two possibilities. If I go to n plus 1 in the first time step, this will happen with probability Tn plus 1 given n. Then, and then go to fixation from there. This will happen with probability pi n plus 1. Right? Or if I go to n minus 1, then I will have to go to fixation from n minus 1. And if I stay where I am, so this is this thing here, T of nn, then I have pi n again. Right? So this recursion relation has to be satisfied. And this thing, as I said, is 1 minus T of n plus 1 n minus T of n minus 1. Okay? And so now we can write this as a recursion, or we can rewrite this in terms of pi n. So we just put the pi n. So we collect, so we have one term here that is proportional to pi n. We can basically cancel, if you will, we could cancel this one against this term, and then we can take this part to the other side. So then we get pi n is equal to to write this in a more symmetric way. Plus minus 1 n minus 1 divided by n plus 1 n. Right, so now I'll just put this over to the other side and divide it by this factor. And now you see that, and this is sort of the nice thing about this, is that this factor here is common to both these transition probabilities, so we can just cancel it out. Right? They are both proportional to this factor, and they just differ by a factor of 1 plus s. So we can actually write this as simply, so here we have, so the sum, so this is proportional to 2 plus s, this is proportional to 1 plus s. So we get something like, is that right? Yes, so we get 1 plus s divided by 2 plus s times pi n plus 1 plus 1 over 2 plus s divided by times pi n minus 1. Okay? Because the sum of these terms is just 2 plus s times this independent thing which cancels. And now we can, for example, solve for pi n plus 1. And this gives us 2 plus s over 1 plus s times, let's put this on the other side, times pi n minus 1 over 1 plus s times 2 plus s. That's 1. And if you now, so now you can sort of recognize that this is equal to, this is actually 1 plus 1 over 1 plus s. And you can subtract pi n from both sides, and then you get this very simple relation, pi n plus 1 minus pi n is equal to 1 over 1 plus s times pi n minus pi n minus 1. Okay. So the difference is, of course, you see that in the case s equal to 0, in the neutral case, these would be the same. So in the neutral case, that just means that the fixation probability is a linear function of n. But here you see that the fixation probability sort of, the differences of the fixation probabilities, they decrease as you go to smaller n in this sort of geometric fashion. So we'll see later, I mean, on Monday, we'll do a kind of continuum version of this, and then this will become a differential equation, which has more or less the same property. Now to solve this recursion relation, of course, we need boundary conditions, or we need, since this is in principle a recursion relation of two steps, we need two initial or boundary conditions. And the boundary conditions clearly are that if I start at 0, then the fixation probability is 0, and if I start at n, the fixation probability is 1. So pi 0 is equal to 0, and pi n is equal to 1. And so this way we can, so this means that, for example, so pi 0 is equal to 0, so pi 2 minus pi 1 is equal to 1 over 1 plus s times pi 1, right? And so let me call is that, okay, maybe I don't even need to introduce this notation. So this, okay, so this is certainly true. And so from there on, we can just iterate this, right? And then we see that pi n plus 1 minus pi n is equal to 1 over 1 plus s. So every time I iterate this, I get another factor of 1 over 1 plus s. So I get 1 over 1 plus s to the power n times pi 1, right? Yes. So this is true for n equal to 1, right? So we have pi 2 minus pi 1. Right, so this is certainly, so this basically solves our recursion relation up to the determination of pi 1. Right. So this is just, iterating this. So every time I do one iteration, I get a factor of 1 over 1 plus s. And pi 1, of course, at this point is still unknown. But now I can just sort of integrate this, right? So this is a kind of difference equation for the pi, which I can integrate. And so this will just give me a geometric series. Is it clear? Okay, so pi n is just the sum of these differences, right? And so this geometric sum I can just do. So this gives me pi 1 times 1 plus s divided by s times 1 minus 1 over 1 plus s to the power n. And at that point I can use the second boundary condition, so that tells me that if I go to the other boundary, so pi n is equal to pi 1 okay, this is not too illuminating. So we have a capital N here, but this has to be equal to 1. Right, so that fixes pi 1. And then we're finished. So the final result is the following. So this is the final result for the fixation probability. Okay, so this is the probability that if I start with small n individuals of type A, and if they have a selective advantage, which is S, that eventually they will go to fixation. Okay? And this is actually a very useful formula, so we will, on Monday, we will look at some other ways of deriving it, but it contains a lot of biology in some sense, or at least a lot of different cases which turn out to be of a certain relevance in the interpretation of evolutionary processes. So let's look at some limiting cases. Okay, we can look at some limiting cases of this formula. So first of all, of course, we can recover the neutral case. So if I take s to 0, then of course you see that the denominator and the numerator, they go to 0, so you have to analyze the leading behavior. But what you find is that this, of course, reduces to what we already know, namely that pi n in this case becomes equal to capital N over small n over capital N. So this is certainly recovered. Then we can look at what happens when the population becomes very large. Right? So we can take the population size to infinity, keeping the number of A individuals fixed. And then you see that you have to distinguish two cases. So if s is positive, then you see that this term here, 1 plus s to the minus capital N, will go to 0, right? So if s is positive, then pi n will become independent of the population size, and it will be 1 plus s to the minus capital N. Right?
X is less than 1 or bigger?
Let me think. I think I don't think there's really a restriction on S. Could be these are sort of relative probabilities. So in some sense, because the probability of choosing an individual is, in this case, WA divided by WA plus WB. So so this will always be between 0 and 1. So S can be, yes, you can take S to infinity. S is anything. So here you see that this so this is of course always between 0 and 1. And a particularly important case that is often considered is when you only start with one individual, right? So if you think about this in terms of mutational events, so suppose a mutation creates an A-type, a single individual with an A-type, what is the probability that this individual will actually survive or go to fixation? So this is pi 1, and so for n equal to 1, this is just equal to 1 minus 1 over 1 plus s, which is the same as s over 1 plus s. And so when s is small, I mean in practice s will often be small. So if you look at mutations that occur, for example, in these microbial populations that I discussed on Wednesday, then s will be a few %. So often one can assume is that s is small compared to 1, and then the fixation probability will be proportional to s. So if you have a mutant which has an advantage of a few %, then the probability that it will actually survive in the population if it appears is only of that order. Okay? But you can also ask what happens if you make this clone. So if you assume that the number of individuals of that type gets bigger. So if we take n to infinity, or make n large, small n large, and s small, then you see that this can be approximated with an exponential function. So this will be something like 1 minus e to the minus n times s. And so what you see here is that 1 over s defines a kind of scale for the population. So this probability, so if you have a small selective advantage, then the size of this clone, this A population, at which it becomes probable that it will actually survive, is of order 1 over s. So a population, so the A population needs to have size, a size of order 1 over s to survive, right? To actually be able to survive with high probability. So if s is 1%, then you need at least 100 A individuals to have a good chance to propagate them in the population. So this is what happens for mutations that have a positive effect. On the other hand, you can also look at mutations that have a negative effect. So let's suppose s is negative. So negative s, of course, only makes sense when s is smaller than, is greater than -1, right? So in that sense, this is sort of not become negative. So when s is negative, and you take n to infinity, then you see that this term will become very large. Of course, when s is negative, then you have to sort of, these terms are greater than 1, so basically you have to write in this form. And when capital N becomes very large, then this will be something like, this is now a term that becomes very large. So this will go to 0 exponentially, right? So this is 1 over 1 plus s. Okay, so maybe I should write it like this. 1 minus s absolute value to the power minus n. So it will be something like 1 plus s, 1 minus s absolute value to the power n, right? And so this will be exponentially small in capital N. Right. So this will go to 0. And so a mutation that decreases the fitness, a so-called deleterious mutation, will not be able to fix in a large population. So maybe this is a good point to introduce those terms. So deleterious mutation means that S is negative. Fix in large populations. And the other case that we discussed down here are called beneficial mutations. So beneficial mutations are those with as positive fix with probability. With a probability that is proportional to S. This is sort of the, when S is small. So this is sort of the main message here, that, and this is the feature that is practically relevant here, which is that if you have a mutation that has a small fitness advantage, it will go to fixation with a probability of order S if it appears in a single mutation, in a single individual. And if it's deleterious, it will not go to it will go extinct, essentially with probability 1. So this is the main point that I introduced from this. Now I derived this here for a very specific model, for the Moran model, but as we will see on Monday, this is actually these features are very robust. So there's a continuum approximation, which is called the diffusion theory, which essentially up to some trivial factors is the same for the Moran model and for the Wright-Fisher model, and within this diffusion approximation, one can derive a result that is quite analogous to this. And this is in fact what is mostly used in practice in population genetics. People usually think in terms of this diffusion process, and there the fixation probabilities have essentially the same properties up to some factors. So for example, we'll see that in the Wright-Fisher model, there's a factor of 2 in front, so rather than being equal to S, the fixation probability is 2S. This is a famous result due to Haldane, but in practice, of course, doesn't matter very much. Okay, are there any questions about this? Okay, so that was a story about the statistical. I have sort of, I don't know, can we go on for another 10, 15 minutes, or do people want to go home and have a weekend? So let me just say one more thing. So this is something that will also turn out to be sort of important in the following. And this is about time scales. So far, we have only discussed how whether the A population or the A mutants go to fixation or not. But we haven't asked how long this actually takes. Right? So the question is, what are the time scales? So let me maybe make a next chapter about this. So this is about time scales. first, so for the first case, let's go back to the neutral situation. And two types. So we just have types A and B, and rather than asking for the fixation probability, let's ask for the time dependence of a certain quantity, namely the so-called heterozygosity. So this is, remember I discussed this on Wednesday. So this is sort of relevant if you look at mating. So you have two different alleles in the population, and heterozygosity is the probability that an individual has two different alleles, right? A diploid individual has two different alleles. Now in this context, this simply means that if I pick two individuals at random, what is the probability that they are of different types, right? So these individuals are just genes in the gene pool. I pick two at random in a mating event. I want to know what is the probability that they are of different types. And the question is, how does this change over time? Okay? So we consider the heterozygosity. So this is a probability that two randomly chosen individuals are of different type. And this is obviously, this is sort of in the neutral, so let's call this H, right? So this is equal to the probability that the first individual is of type A times the probability that the second individual is of type B, or vice versa, because we have this possibility of doing, of the order, there's a factor of 2, right? So this is obviously equal to capital N over small n divided by n. So this is sort of the value of the heterozygosity in the population, right? Now, you might think that because capital N, so we know, this is this martingale property, that if I now propagate this, for example, according to the Wright-Fisher model, that the average value of small n will not change, right? So you might think that this also means that the heterozygosity doesn't change, but this is not quite right, because the heterozygosity is not a linear function of small n, because it contains also the second moment, right? So if we now want to calculate the change in the heterozygosity, so the change in H. So let's sort of take one step in the process, that we call this H prime. So this is the expected value of 2 times n over n times 1 minus n conditioned on n, right? And what this actually requires is then the not only the first moment of n prime, but also the second moment, right? So this is basically equal to 2 times 1 over n times the expected value of n prime given n minus 1 over n squared. This comes from the second term here, times the expected value of n prime squared conditioned on n. Alright. Now what is this? I mean, so this of course now depends on the model. Now in the Wright-Fisher model, let me just write this here. So in the Wright-Fisher model, because you're doing binomial sampling, the variance of n prime conditioned on n is just equal to n times PA times 1 minus PA, which is equal to n times. Okay, so this is the variance of n prime, and from this you can calculate the expected value of n prime squared. And so the upshot, I mean, this is a fairly trivial calculation, but the upshot of this is that the heterozygosity after one step decreases by a factor of 1 minus 1 over n. Okay, this is easy to work out using this relation. And so the heterozygosity actually decreases. And this is sort of in population genetics, this is sort of a standard way of quantifying what people call genetic drift. So the fact that the sampling, this random sampling, reduces the diversity. And so here you can sort of see this in an average way. So rather than just looking at the endpoints of fixation, here you can see that during the process, the heterozygosity actually decreases. So after t steps, if you do this after t generations, you see that the heterozygosity after t generations is essentially the heterozygosity at time 0 times 1 minus 1 over n to the power t, which you can of course write as an exponential function essentially, right? So you see that when n is large, that there's a time scale 1 over n. So 1 over the population size, or the t over n, I'm sorry, t over n. So there's a time scale equal to the population size on which the diversity decays. Right? And this is called the time scale of genetic drift. So there's a time scale. And so this also means, I mean, this I haven't of course proved with this argument, but this also means that if you now look at this process of fixation, and so you know that eventually one of the two types has to go to fixation, and you can ask what is the typical time until this happens. This time scale will also be of order n. Okay, so there's a time scale of order n until fixation occurs in the neutral case. In the neutral case. Now just two more remarks about this. You can do the same calculation for the Moran model. So the Moran model. Now the Moran model, of course, is a little bit artificial in the sense that you only, in each, I mean, you don't really want to call a time step in the Moran model a generation because you only reproduce one individual. So in some sense, it's obvious that in order to get a full generation, you should actually do n time steps. And correspondingly, in terms of Moran model time steps, this fixation time is bigger by a factor of n, right? So this this time scale. But there's also a factor of 2, which always causes a sort of confusion in the literature. So let me mention that here. Equal time scale is t is equal to n squared over 2 in units of reproduction events. Right? So that it's n squares, as I said, is a kind of trivial effect of the fact that you only reproduce one individual at a time. But there's also this factor of 2, which just comes out of the calculation. And the second remark that I want to make, that if you now, of course, you can ask what happens if you have selection, and then the time scale is much shorter, as you might think, because the process is sort of driven towards one of the boundaries. And it only depends rather weakly on population size. And it turns out, and this is a calculation that is not so easy, and I don't want to do it here, it's logarithmic, so it's inversely proportional to the selection coefficient, and it's logarithmic in the population size. And that, I think we will make use of that later on, so I just wanted to mention it. And I think you will find some more details about this also in the lecture notes. I will send them around. Okay, so I think that's information. So on Monday we'll meet at the same time and continue with the diffusion theory. Thank you.